In this video, we want to look at ways that we can manipulate DNA. Our ability to manipulate DNA and use it in the laboratory has given rise to tremendous possibilities that now exist in many different fields. For instance, you're in biochemistry right now. Definitely our ability to manipulate DNA affects biochemistry. It stretches across many disciplines like cell biology, genetics, medicine, pharmacology, etc. And so it is very possible that you use products or you will use products in the future that have arisen because of manipulating DNA. And so because it is such a common thing now in science and society, we want to be familiar with the common techniques that are used. And so most of us, if we start down at the bottom of this picture, most of us are familiar with ways or products that use DNA manipulation. For instance, most of the food that we eat actually can be genetically modified to contain genes that make the organisms resistant to certain pesticides or even different insects that might eat the plants. We see a lot of genetically modified organisms in the basis of plants that are used for a lot of our food products like corn and soybeans. It's been a buzz for a while that we can perhaps manipulate the genes in bacteria and perhaps release those bacteria then into the environment if they can metabolize certain toxic wastes that might help us to clean up different types of chemical spills. And in medicine, we use proteins actually. The body's own proteins, we can take them and use them for different types of therapies. So for instance, part of the clotting pathway that allows clots to dissolve once they're formed, we can now take those proteins that the human genome encodes for, and we can produce them in vitro. Same thing with different hormones that are released by the endocrine system, like human growth hormone. We can now produce that in a lab and give it to patients. And even more common than that example would be insulin. And so many diabetics, both type 1 and type 2 diabetics, take insulin, which is a protein. And now, instead of deriving that insulin from animals like pigs, we can produce it, human insulin, in a lab. And so if we look then how we got to these outcomes, it was because we knew how to manipulate DNA. And so what we can do now is take a gene of interest from some type of cell, and we can take that gene and place it into what we call a plasmid or a vector. These vectors then we can place into cells that can replicate quickly. And so as the cells replicate or divide, with them will go that gene of interest that we are looking at. And then that gene of interest, we can either do genomic research with it and put that gene into organisms, or we can use these cells that are producing the gene of interest, and we can have them continue with transcription and translation, and then we can do research or use that protein for treatments. So the steps of what we just saw to be able to use DNA and manipulate DNA, to pull it off, the first step is being able to put our gene of interest, or to put that DNA into an organism then that can either replicate the DNA or make subsequent proteins with it. So many times when we start to manipulate DNA, you'll see that we use cells that can divide very quickly, such as bacteria, and the most common one is E. coli bacteria. Or we can use eukaryotic cells if we want to have our end protein have certain modifications on it that bacterial cells do not do. So we can, for a eukaryotic cell, use yeast cells. And so how we start is we have to make the plasmid that will carry the DNA. So a plasmid is a circular piece of DNA that is very simple, and it will have in it restriction sites that we can cut with our restriction enzymes, and we can place our DNA into that plasmid. Another name for a plasmid is a vector. Generally, when we cut our DNA and our vectors, we have to use our restriction enzymes, and those give us sticky ends to our DNA. And so to place the DNA into that plasmid, we have to use another enzyme called DNA ligase that acts like glue, and it will literally glue those sticky ends to our DNA into our plasmid. Once we have our plasmid made, remember another name for that is a vector, we can put that vector into cells. And when you put a vector into a bacterial cell, we call it transformation. We can also use viruses, and we can use viruses to put the gene of interest into a cell, and they do that by infection. And so by creating our plasmid, we can then put it into cells to either have those cells increase the amount of the gene by DNA replication, or by transcription and translation produce RNA and then protein. If we look at the whole process, we can see that there's many important steps that you want to know and be able to highlight. And so we start with a plasmid, and these are created in the lab, and they will have many different restriction enzyme sites in them. And so what we can do is we can use the same restriction enzyme, and we can cut this plasmid, and then we can cut our gene of interest. And as we do that, you can see there are sticky ends that are created on our DNA of interest, and those are going to match precisely to the plasmid if we use the same restriction enzyme. They'll find the same palindrome and cut those nucleotides in the same exact way, so that then they will anneal together. And that annealing takes place with an enzyme called DNA ligase. It literally glues that DNA of interest into our plasmid. Once we have our plasmid created, we can then place our plasmid into a cell. And like I said, we can do that by using certain chemicals. When we do that, it's called transformation. Or we can put that plasmid into a bacteriophage or another type of virus like an adenovirus, and then we can use that virus to infect many different cell types, and as it infects it, it will pass on our gene of interest. And so however it gets in there, once that plasmid carrying our gene of interest is in our cell can go ahead and divide and divide and divide. So as the cells divide, they will be replicating that plasmid DNA, and as they replicate that DNA, will increase the amount of DNA in the cell. There are some types of bacteria that will actually be able to, once a plasmid is in it, the plasmid contains regulatory sequences that will have this plasmid replicated continuously. And so you can have a plasmid that will have you can have a bacterial cell that can have many plasmids produced in it. And we're talking on the scale of like hundreds to thousands. When we're ready to use our DNA, depending on what we want to do with it then, we can then take these bacteria, lyse them, that means break up their cell membranes, and then we can extract out our DNA of interest again if we were interested in the DNA. If we look at a plasmid or a vector, they have multiple restriction sites. So each of these letters that you see here, those are restriction sites, and specific restriction enzymes will cut them and create sticky ends. The other thing that is on here in these colored areas, these are what we call selection markers. So these are genes that have, or they create, their gene products, create enzymes that will allow us to identify cells that have the plasmid present in them. So for instance, here is the gene It's called ampicillin resistance gene, so you can see AMPR. If a bacteria has this plasmid in it, and this plasmid is undergoing transcription and translation, this plasmid gene right here, this AMP gene, will create an enzyme that allows the bacteria to be ampicillin resistant. So it will metabolize the ampicillin antibiotic that is put in the media, and these cells that carry this plasmid will then be able to grow in the presence of the antibiotic ampicillin. Over here you can see this LAC-L and LAC-Z. So the LAC-Z gene carries an enzyme called beta-galactosidase. And beta-galactosidase is an enzyme that can cleave what we call XGAL. So here's this XGAL molecule. If beta-galactosidase is present, it cleaves that enzyme, and when it does, one of the metabolites has a blue color, only if it is cleaved. And so you can see the beginning XGAL is a colorless compound. When it's cleaved by beta-galactosidase, the metabolite turns blue. And so bacteria that have the plasmid present, they will have a blue color if they are plated on agar that contains XGAL. Here's an example of that so you can see it. So each of the little dots is a bacterial colony, and the thought is that each colony is arising from one bacterium, and that one bacterium passes on its genetic information to all of the cells in that colony. And this is a XGAL plate, so in the agar there's XGAL present, and so only in colonies that make beta-galactosidase, those colonies turn blue and we could select them. So these blue colonies contain in them the vector that encodes beta-galactosidase. So if that was our gene or marker of interest, now we know the cells that would have our piece of DNA that we are interested in. And so, like I said, this is a very common system, the XGAL beta-galactosidase system, but we can do the same thing with antibiotics. So these plasmids in biotechnology have a very important role. We can use them to express recombinant proteins. Then with those proteins we can use them to treat different pathologies or use them for basic science. We can clone genes, so we can have multiple copies of a gene to manipulate it further. Or we can use plasmids to house whole genomes or many different types of cloned genes. And when we do that, it's called a library. And so the concept is the same as a library that you would go to study in. So a library is a building that contains many, many volumes of books. And when we think of a genome houses many, many, many different types of genes. And so an analogy that's always given is a genomic library. Instead of having a building with tons of books, instead it's many plates of bacteria where each colony is like a volume. Each colony is like a single book in that it houses and contains a gene. And so genomic libraries can be one of two different things. They can either be what we call a genomic library, and so genomic libraries house all of the different components to a gene. So we're talking regulatory areas, introns, and exons. You will also hear of cDNA libraries. So cDNA means complementary DNA. These complementary DNA libraries are actually made from messenger RNA. The messenger RNA gets reverse transcribed back into DNA, and so then that DNA is the segment of the gene that only encodes for the protein, and so it does not have introns and exons. Either way, how you make a genomic library is very similar. So you take your DNA of interest, so here we're doing it with human DNA. We use multiple restriction enzymes to cleave that DNA into millions of different fragments. We then use those same steps of creating a plasmid to create many different types of plasmids, and then we transform bacteria so that our bacteria clones will each have a plasmid. So you can see these are each like a book in a library. They each contain information in them. So for this to work and for us to be able to clone and create a genomic library, we want to make sure that we have all of the genes that would be present in an organism. And so to ensure with high probability that a desired sequence is represented at least once in the library, there has to be a high number of randomly generated fragments. And so this is just subject to the laws of probability. In your textbook, you'll see that there's a formula, and we can figure out how many fragments or then how many clones of those fragments you need to be able to ensure with about 99% certainty that a particular size fragment is represented in your library. So for instance, put some numbers to that. For the human genome, we need about 1,400,000 clones are needed to be about 99% sure that we can include genes of a particular like 10,000 base fragment or base pair fragment. Once we have our library, we need to be able to identify the bacteria in the colonies that might have our gene of interest in them, right? So a library is no good if you cannot use it, if you cannot be able to find the book that you want to read. And so we have what we call in situ hybridization. It's a common molecular biology technique, but what it does is it helps us to identify bacteria DNA that would be of interest to us. And so here you can see is our colonies, and this is a master plate. So you never want to have to go through all of these steps multiple times when you want to find your DNA. And so what we do is we keep a master plate, and this can be kept in a refrigerator. When we're interested in finding our particular DNA sequence in the library, what we do is we can get basically a stamp. And this has velvet on it, so a soft surface that's not going to disrupt and break all of our colonies. And we stamp our stamp into the colonies, and part of that bacteria will be transferred onto our stamp, and this one has velvet. We then take our stamp and we stamp it onto nitrocellulose, so that's like a paper filter that will anneal the DNA. And we can then get our bacteria. We lyse the bacteria so that we're left with DNA bound to our nitrocellulose. Then we can use probes, and so to be able to find your colony of interest, where your DNA is located in the library, you have to know something about the gene of interest you're going towards. So you have to know a couple of the nucleotide sequence of that gene of interest. And normally what we can do is make very small radioactive probes, and then we can get our nitrocellulose filter. We can anneal our probe to it, and we can incubate it, and then we wash off all of our labeled probe, and then we can take radioautograph film, and we can place it onto that filter, and when we expose the film, we'll pick up on the radioactively labeled areas, and that will point us to the area on our original plate where we have that gene of interest located. So then we would be able to take that bacteria and we could grow it up knowing that we have our gene of interest. So once we have located our colony of interest, we would then go through all of the steps again to grow up our bacteria, to amplify that DNA if that's what we want. and then we would be able to use it for all the other applications that we have with our recombinant DNA.